All right, we have Dr. Martin here this morning with us, and he's going to talk about a subject near and dear to his heart and individual tree growth.
All right. Hey, everybody. Yeah, I think the idea here was to give you all a really brief refresher or primer on how trees grow, because obviously that's really what we're measuring when we go out and do a timber inventory, for instance, or an inventory for forest productivity. We're trying to measure the growth of a forest based on our measurements of individual trees. And like most things we do, it's helpful if we understand the underlying mechanisms of what we're measuring. So I think that's the idea here. I looked at the attendance list, and a few of you are in my tree biology course right now. And so this will be a bit of a review for you, but I don't think that's a bad thing. I think, especially in this context, this will give you the opportunity to think about what you've learned relative to how we measure trees. So let's jump into it. I think a good place to start is to think about how the growth of trees differs from the growth of other organisms. Can anybody, offer a explanation of how those two things are different? I'm patient. I can be quiet longer than you can.
Be the types of fuels that they use to grow.
Yeah, that's great. Um, Plants are autotrophic, and they generate their own food through photosynthesis. So that's that's definitely a difference. Um, in terms of a little bit more of the nitty gritty of how they grow, are there any other differences you can think about how you or I grow versus how a tree grows?
There's a comment in the chat from Ethan.
Yeah, he says trees grow from the top with apical meristems.
Okay. Yeah, Ethan is in class in tree biology class this semester, so that's gratifying to see a tree biology person jumping in there. And he's right. Organisms like animals, mammals, and most animals grow everywhere. So this is a growth progression, a chronosequence of my now 13-year-old twin boys from near birth to recently. And as they grow, cell division occurs all over their body. Uh, cells in their liver divide and their livers get bigger, and cells in their bone plates divide and their bones get longer, and so on. So cell division occurs all over their body. Whereas in plants, growth is more localized. Growth is localized in tissues called meristems. So that's a real fundamental difference in growth between animals and plants. Um, so in plants, growth occurs in meristems, and meristems are just regions or tissues that have rapidly dividing cells in them that produce new tissues and also reproduce themselves. So that's the definition of a meristem. And there are two broad classes of meristems. There are primary meristems and secondary meristems. So primary meristems are also called apical meristems, And those are the growing tissue or the meristems that produce growth in length or height. So like Ethan pointed out, growth of trees occurs from the top because the meristems are located at the tips of all the branches and roots in the tree or in the plant. So if we think about, the bud, when we grab a branch and look at the tip of all the branches, there's a little bud there. That bud contains the apical meristem. So that's where growth is occurring. For growth in height or length, that's where it's occurring on trees and plants. This is sort of a close-up. This is not a tree. This is some other plant in a textbook, but it shows you basically where cell division occurs in a plant, and the lines indicate zones of rapid division. So the apical meristem is at the very tip of the plant. Now, also note that there are some regions further down on the stem of this plant that also have some cell division occurring, and those regions are called intercalary meristems. And basically what that means is that the apical meristem has produced all of these tissues, and those exist in the terminal bud. But when that bud expands, it's not just the apical meristem itself that's producing shoot expansion. There also are a few more zones, basically in between nodes, that are also generating additional tissue and allowing for the expansion of that shoot. So that anybody that's seen an expanding pine shoot has seen this happen. So this is a pine shoot in the spring when it's first expanding rapidly. And what happened was basically, see all of these needles? All of those needles existed inside the bud as tiny little baby needles, and they overwintered. So this first flush in the spring, there were little tiny parts that overwintered, and when the bud expands in the spring, the apical meristem has already produced all those little baby needles and shoot parts, and there are intercalary meristems between each needle that then expand that shoot. Okay, so this shoot expansion is basically the result of the action of the apical meristem and also those intercalary meristems. But those are all primary meristems. They produce growth in length or height. Okay, so we are seeing this happen right now. If you go outside and take a look, especially at a pine shoot, you'll see these candles. That's just rapidly expanding shoots. And there are different patterns of shoot expansion. I'm not going to go into them in this lecture, but pines, southern pines, for instance, have several cycles. They can have 2 or more cycles of shoot growth during the year, where basically you see this candle expansion throughout the year. So pines are cyclic. There are other species like sweet gum, for instance, that just continues producing new shoot parts and expanding throughout the year continuously, indeterminately. And there are other things that are in between. So there are different shoot growth patterns, but the takeaway here is that it's the action of that apical meristem that causes growth in height of the tree or growth in length of branches and roots. So, that sort of leads to this classic botany question. So someone hammers a nail into a tree and hangs a lantern on it, and then comes back 10 years later, and the tree has grown in height. So where will the lantern be after 10? A or B?
Same spot.
Same. Why?
Because it's only growing from the top.
Yeah, because growth in height of the tree occurs from the tip. The apical meristems are in the tips of the tree. No growth in length of the stem occurs down here.
I had that same question posed to me in school over 45 years ago.
I think we all did. You know, it's either it's a hammock or a lantern or whatever. And, you know, we things on the internet. Here's one that I actually collect these. This one said, a boy, before he went off to World War I, he leaned his bike against the tree. And then when he came back from war, the bike was 10 feet up in the air. Not possible. What do you think happened here? How did that happen? How did that bike get up in the air like that?
It could have been placed in a fork in the tree.
Yeah, exactly. There was a fork. You can sort of see the fork there. Someone put the bike up in that fork, and the action of a secondary meristem, which I'm going to talk about in a minute, the growth in diameter of the tree basically enveloped the bike as diameter growth in the main stem and in that forking branch occurred. We see that with fences, when someone hammers a piece of fence onto a tree. So can anyone think of plants where the apical meristem is knotted at the tip, like in trees?
Grasses.
Yeah, grasses. I didn't hear who said that. I wonder if it was someone in tree biology.
It was Ethan.
It was Ethan. Good job, man. Yeah, so grasses, many or all grasses that I know of, their apical meristem is at the base, at the bottom. So they do grow from the bottom. What would be the evolutionary advantage of having your apical meristem at the bottom?
If you keep getting chopped down, you can grow back.
Yeah. If, a grazing animal comes along and munches your vegetative part down to the ground, the animal hasn't eaten your apical meristem, and you can still continue growing. Or if a fire comes through and destroys the upper part of your body, your apical meristem is down on the ground, is a little more protected, and growth can continue rapidly instead of having to regenerate a new apical meristem from a dormant bud or something like a tree would have to do. So yeah, great. So obviously then differences in plants sometimes about how those apical meristems are configured, but in trees they're at the tips of branches and roots. The action of these apical meristems is sometimes under control by hormones, and that's one of the things that results in the distinctive appearance of different trees. So, for instance, two broad classes of trees are Xcurrent and Dcurrent crown shapes. So, sort of classic Xcurrent trees, sort of Christmas tree shapes would be trees like a lot of true firs or spruces. Young pines have a pretty Xcurrent kind of crown form. And that is caused by suppression of growth of the apical meristems in lateral branches by hormones in the terminal bud. When that influence is strong, it's called strong apical dominance, and an Xcurrent crown form results. On the other hand, some species, like this one here, that influence of the terminal bud is not strong, and apical dominance is weak, and basically all the lateral branches grow pretty much the same rate and also grow up towards the sun. That's an example of weak apical dominance, and the result is a crown form called Dcurrent crown form. That also then reminds me of a slide that I forgot to put in to this lecture, but we are in an adaptive time, so we will adapt. So let's say we've got a tree, my awesome art skills here, and that tree is growing, and it has strong apical dominance. So it sort of has that Christmas tree kind of form. And let's say that a windstorm comes along and kills that terminal bud. Can anyone hypothesize what might happen if this tree that has strong apical dominance, if the terminal bud gets killed? What happens? Maybe someone that's not in tree biology could give me a hypothesis.
Well, in longleafs, doesn't it, if the fire kills the terminal bud when it's in grass stage, it'll die?
That can happen. You know, certainly, you know, a longleaf, basically the terminal bud is its only apical meristem above ground, and if that is killed, that certainly can kill the seedling. So a longleaf seedling or any other tree seedling that's really small, if its terminal bud gets killed, it could certainly be killed.
Would that create a fork where one of the intercalary nodes would take over at that point, and then it might start branching off in a new direction?
Excellent. Yep. So Chris is right that especially in a tree with strong apical dominance, if the terminal bud gets killed or damaged, basically it's not producing those hormones anymore, and the apical meristems in lateral branches no longer are being suppressed by those hormones, and they will start growing more rapidly and start growing up towards the sun. Okay? If they sort of grow at similar rates, and they continue growing upward, a fork is produced, and you have a fork in the tree crown. So, you know, this is certainly something we see in inventories all the time, right? When we look up in tree crowns, we see forks, and that's how they occurred. If you look up in a stand and you say, wow, there's a lot of forking in this stand, and they're all at about the same height. It looks like most of these trees are forked at about 25 feet. That's a little clue that maybe there was a disturbance that moved through that stand when those trees were about 25 feet tall, and a lot of apical meristems were damaged, and that triggered a lot of forking in that stand. So that's sort of a clue we can get. In some cases when this happens, you know, one branch might end up growing faster than the other, and it basically wins the race, and then this branch begins asserting apical dominance over even the other lateral in the same whorl, and it continues growing up, and it becomes the main apical meristem of the tree. And let's say, you know, diameter growth continues, and, you know, maybe 15 years later, that tree I don't know how to do this. That tree ends up looking like this. What do we call that?
Sweet.
Yeah, it's sweep or a crook, you know, where basically there's a detour in the stem. Same thing. It happened when apical dominance was lost because of damage to the terminal bud. A lateral took over, but there was only one lateral that ended up winning the race, and that results in in a crook in the stem. So just some examples of what you might see in the woods.
Tim, a short comment is that in newly planted stands, depending on the area, and I haven't seen it in quite some time, we can get damage from browsing, essentially from rabbits, sometimes deer, but not so much deer, but rabbits have been known to really do a lot of damage to a very newly planted stand, and we get this situation later on in the life of the.
Yeah, that's a great example. And like titmoth can be a
another
big
good one.
problem in loblolly pine, especially sort of during when they're saplings, and it can cause this. On the other hand, sometimes it happens early enough that the lateral that takes over really isn't offset much from the stem, and you would never know it, a few years later. So this certainly can happen, and it may not be detectable. That crook might just be a slight departure that gets covered up by secondary growth. So there's lots of different ways this can manifest. So yeah, that's a great example, Russ. Okay, a little detour, but I think that's useful information. Okay, so that's primary meristems, growth and height. Secondary meristems are also called lateral meristems, also called cambium. That produces growth in width or thickness. There are a couple of kinds of cambium. The vascular cambium is the one that produces the vast amount of growth in width that we see. That's the tissue that produces xylem to the inside and phloem to the outside. There's also cork cambium. That's the tissue that produces the outer bark, the corky protective layer of bark on the outside. So that's a separate meristem that produces that. The vascular cambium is like a sheath that encloses all of the stem, branches, and roots. Okay, they all, if that organ lives more than a year, they all have a cambium that produces growth and width. So roots can have secondary growth and width as well. You can even see growth rings in roots. Okay. So like this is a cross section of a tree stem that the vascular cambium, they've peeled away the outer bark, inner bark, and phloem. So this surface is the vascular cambium. So the vascular cambium is producing the phloem to the outside, the xylem to the inside. Okay? Let me show a quick animation of how that works. Okay, so this green zone is the vascular cambium, and it just produced cells to the inside that are going to become a xylem cell.
I like that little clock there that shows what time of year this is happening.
Yeah, this is a nice animation. One thing to point out that I didn't mention before, he talked about the production of another layer of phloem to the outside, and then old phloem is crushed against the outer bark. That crushed inactive phloem is what we call inner bark. So inner bark is actually not true bark. It's dead, inactive phloem that's just been crushed by the production of new active phloem to the inside. Okay. So that multiplicative division, there are some cell divisions that go this way instead of this way. that just produces new vascular cambium. Obviously, as the tree gets bigger in diameter, there have to be more cells to reach all the way around the tree, right? So just to clarify, they kept talking about division number 1, 2, 3, 4, 5, 6, 7, 8. That's just for illustration purposes. If you counted layers of cells in a tree ring, likely there would be, scores to hundreds of layers of cells that are produced in a growing season, but they just had 8, for illustration purposes. Also note, he mentioned the difference in early wood and late wood, and we'll talk about that here next. Any questions about that animation? Okay, so secondary growth, that's what produces the tree stems. So clearly, secondary growth in trees is the basis of forestry, obviously. That's what gives us wood. Wood is just xylem, okay? We chip off the bark and inner bark, and we use the xylem part for most wood products. Obviously, we use bark for mulch and stuff like that, but that the xylem is really where the main products come from. So some trees can get really big. This is in northern Georgia. This is Cove hardwood. I forget exactly what it is. It's like a buckeye or something like that, but it's a gorgeous one. So like was mentioned in the video, there are patterns of cell production during a growing season in temperate trees. So early in the growing season, those xylem cells produced early in the growing season are called early wood. This is why most people do well in tree biology, because most of the concepts are pretty straightforward. Early wood is produced early in the growing season. It has different characteristics than xylem formed later in the growing season usually. So early wood or spring wood tend to have thinner cell walls and lower density than wood produced later in the growing season, where often the xylem cells have thicker cell walls, smaller holes or lumens in them, and that's higher density. Okay, that alternation between early wood and late wood is what gives the appearance of growth rings that we see. So growth rings are just the result of alternating bands of early wood and late wood. Now in different species, that appearance of early wood and late wood can differ. It's usually pretty obvious in most, for instance, conifers that we work with. Some hardwoods or angiosperms look a little different, and I'll show you some examples in a minute. The difference in density between late wood and early wood means that the ratio of those two ends up being pretty important for the density of wood on average in the tree stem. If you have a tree that's producing a lot of late wood relative to early wood, then the wood that comes out of that part of the stem will be denser than, for instance, a part of the stem or a different tree that produces a lot less late wood relative to early wood. The density of late wood and early wood are relatively constant, so it's the ratio of those two that determines the density of wood. So that's an important thing that people measure when they're measuring wood properties, for instance, is the relative amounts of those two. So this is a micrograph of a pine. This is red pine. And what we're seeing here is the tree is growing in this direction. So the vascular cambium was out here somewhere. All of these cells are tracheids, water conducting cells that were produced by the vascular cambium. These cells with the bigger holes in them, that's early wood. And then later in the growing season, you see how the holes are smaller and the cell walls are thicker. That's late wood. Okay. And then at this point, the tree stopped diameter growth for the year. And then the next year, it started producing early wood again. Okay. So that will show up as a growth ring in a cross section or in an increment core, for instance. Now, not all trees look like that. This is an example of an oak. This is red oak. In this case, the tree is growing in this direction. And in trees like oak, which are ring porous species, they produce huge vessels early in the growing season, and the early wood has these huge vessels. And then there's a pretty rapid transition to much smaller vessels. So here, these are the same types of cells as these, they're just much smaller. So this is early wood, this is late wood, and then here's where the tree stopped growing for the year, and then here's early wood the next year. Okay. So obviously the morphology of red oak looks a lot different than the morphology of pine, but it still produces what looks like a ring for a single year's growth. This is another example. This is a diffuse porous tree, which means the difference in size of xylem cells from early wood to late wood is not as abrupt. It doesn't change as much. So this is big leaf aspen, I believe is what that is. Yeah. You can see the cell sizes are pretty much the same throughout the growth ring, and they're a little bit bigger in the early wood, but not much. So in this case, really the main thing differentiating one ring from the next is basically just sort of a line of parenchyma cells between successive years of growth. So there are differences in how we can distinguish growth rings depending on the species and the morphology of the xylem. And things like pines, like I mentioned, it's really obvious that early wood, late wood is usually pretty distinct in pines. Some hardwoods, it may be less distinct. In other parts of the world, like the tropics, there may not be growth rings. Most of you have probably used an increment borer to pull an increment core out of a tree and look at those growth rings. So we can detect those zones of early wood and late wood in cores that we pull out of trees and use those, for instance, to make a rough estimate of the age of a tree. So those tree rings can be informative. Sometimes we have to be careful. So if you're just doing sort of a routine inventory and you don't have access to the records and you need to know approximately how old a plantation is, this is a perfectly legitimate way to do that. If you can hit the pith with your increment borer and count the number of rings, that's a rough estimate of the age of the tree. It's important to remember that depending on where you take that core, you're missing, growth rings that were made before the tree reached the height of your increment borer, right? So you may have to add a year or two for that. It's also important to remember that sometimes, depending on conditions, there may be dropped rings, which means there may have been a year when a ring was not produced, so you might miss a year. Sometimes there are false rings, which means a tree produces summer wood because there's a drought or something in the middle of the growing season, and then it starts raining again and it produces early wood and late wood again. So there may be a false ring, a second ring produced in a year. That can sometimes throw you off a bit. Again, for rough estimations, that's not a huge deal. But if you're doing research, for instance, you're interested in correlating weather patterns over a long period of time with tree growth derived from tree rings, that's a whole nother thing. There's a whole field called dendrochronology that has statistical tools that you can use to deal with things like dropped rings, false rings, and so on. That's a whole nother endeavor. Certainly it's doable. There are good tools for doing that, but coring a few trees here and there and then trying to correlate it with weather in the past can be a little iffy without using some of those tools. Okay. Any questions about tree rings or secondary growth?
Tim, I'm just going to ask one question. As climate change is occurring, the warmer temperatures will benefit our southern pines by producing denser wood?
It's hard to say. Our models do suggest that, especially the elevated CO2 that's causing climate change is likely to cause especially well-managed pines to grow faster. How that will impact wood density is an open question. If we look at, for instance, studies of fertilizer effects on growth rate and wood quality, most of those in southern pines tell us that there are not huge impacts on wood quality, that those early wood to late wood ratios stay approximately the same with increased growth rate, and so wood quality is not enormously impacted. So my first order guess would be that it won't impact wood quality in a huge way, but I'm an academic, so I'll say, well, I don't know. But that's my first guess.
Thank you.
Yeah. Trees aren't cylinders. So I think we all know this. But I just wanted to remind you that, trees have taper. That's because, one, there's more rings at the base of the tree than there is at the tip of the tree. So there's naturally going to be taper. But there's also variation in growth rate in diameter throughout the stem of the tree. And that also will influence taper. So how cylindrical versus how tapered the stem is. And I assume that your instructors, will have or have already had good lectures on how to quantify taper and what form class is and all of those kinds of things. And that's really important, obviously, for estimating stem volume, for instance, the volume of wood in a stand. All I want to say is that taper exists, and it's influenced by a lot of things. It's influenced by stand density, it's influenced by crown length, which obviously is also influenced by stand density. It's influenced by tree age, it's influenced by genetics. So there's a whole basket of things that cause trees to be more or less tapered, which is why, in some cases, it's important to actually measure form class if it's really important that we get it right, for instance. There are some general patterns. In general, dense stands have less taper than more open-grown stands. In general, trees with short crowns have less taper than trees with longer crowns. In general, older trees have less taper than younger trees. The problem here is that, each of these is just one factor. All of these factors interact. So old trees that are grown in dense stands will have different relationships than old trees that are grown in the open. So, these are just general patterns. You can't take any one of these to the bank. But I just wanted to throw it out there that, like everything else, this is biology, and there's a lot of factors that influence taper. Okay, wanted to move on beyond those patterns, talk about the timing of growth. Timing of growth and the timing of biological processes is sometimes called phenology. So temperate trees in temperate zones where we are have a defined growing season, at least the above ground parts. Usually the initiation of growth happens sometime that's around the time of the latest average frost. So this is an adaptive thing. Trees have evolved to where they tend to start growing a little bit after that latest average frost, and they stop growing a little bit before the average earliest frost in the fall. What that means, obviously, is that trees that are adapted to more southern climates have different initiation and cessation dates programmed in genetically than trees that have evolved further north, even within the range of a species. So for instance, loblolly pine from Marion County will have different patterns of growth, initiation, and cessation than trees or seed sources from way further north in the loblolly pine range. In tropical regions, growth may continue continuously throughout the growing season, or may start and stop depending on, for instance, drought. There might be a dry season that doesn't necessarily coincide with what we would think of as winter. So tropical trees behave differently because there aren't as strong seasonal effects on growth phenology. Here's just an example for loblolly and slash pine in a study here in northern Florida. So we measured growth over the growing season for 2 years. In 2002, for instance, we found that growth started on March 10th and ended on October 28th. Whereas the following year, growth started a few weeks earlier and ended a few weeks earlier. So there's some variation from year to year. There's different signals and patterns of temperature that influence when trees start and stop growth. It's also likely that, for instance, water availability may influence growth cessation in the fall as well. So again, there are a lot of factors that can influence that, but there is a genetic component as well. These were mainly trees from the southern, slash pine, is from the south. The loblolly pine source was a southern source as well. These are some data from South Florida slash pine growing near Fort Myers, and these are patterns for 4 different years. The y-axis is not cumulative size like it was in the previous graph. This is just growth rate. So this is like growth rate over that measurement period. And what you see is that these South Florida slash pine are growing all year. There's some diameter increment going on, even in, January in these trees and in December. It's small, but there was measurable growth. Okay. So that's an example. Fort Myers is subtropical, right?
Well, that takes me back there to Gordon Langdon's stuff.
Yeah, I'm dragging out some oldies but goodies here, right?
It was in Lehigh Acres when there was a U.S. Forest Service experiment station there. So
Did you know him?
I met him at the end of his career, in the early 80s.
That's great.
Also, I posted in the chat, there's a national phenology network out there, and you can become an observer in their nature's notebook. So you can actually pick a plant or a tree somewhere that you see all the time and put in observations in there all throughout the year about when it flowers, when it buds out, when different things happen to it. So it's kind of cool.
Yeah, that's cool. And that's actually a neat point, is that some of the earliest solid data about the effects of climate change are phenological data, because people have been writing down when plants flower and start growing for hundreds of years. So we actually have really good records of phenology of plants going back a long time. So we were actually able to detect climate change effects pretty early on, before some of the other more subtle effects of climate change. So this is just some examples of shoot growth phenology in slash pine. again, it's a temperate species. It grows, starts growing in the spring, stops growing in the fall. Whether diameter growth starts before or after shoot growth varies. I couldn't find sort of a generalization. I think in some species, one starts before the other. In some species, they start about the same time. Okay, but in general, phenology, they start about the same time. One may be, a few days or a couple weeks ahead of the other. Just a little factoid. There are also really small variations in diameters of trees that occur daily. So tree stems shrink and swell on a daily basis. So these are fine increment measurements of tree diameter, you see, over individual days. So you can see that during the days trees shrink, and then in the evening they swell up again. And that's associated with water movement and water stress. These fluctuations are really small, so this whole axis is 1/3 of a millimeter. So these are really small fluctuations. We wouldn't detect them with a diameter tape, but it's happening. Okay. Growth patterns change with age. The generalized form of growth of most plants versus their age looks like this. It's a sigmoid or a logistic type shape. And what that means is there's a period of really rapid growth when the plant is young or medium aged. And then as the plant gets older, its growth slows down and its increment is smaller. We design management regimes around this pattern. Generally, if we're growing trees for wood, once they've reached the slowing down point, there's not much economic sense in holding them much longer. From a measurement perspective, regardless of why we're measuring them, this tells us something that, for instance, if we're doing repeated measurements on permanent plots, if we're measuring permanent plots and the trees are in this zone, what's a potential problem that could happen if we went out every year to measure them? What's a potential problem associated with this?
Mortality.
Certainly mortality can occur, and we need to adjust for that in our inventories and in our models. How are we measuring growth with permanent plots? How do we use those data to infer growth? So if we measure a permanent plot at age 55 in a stand, and then we go back at age 56, how do we get growth from those two things? Just in general.
Basal area or diameter?
Exactly. Difference in tree size between those two measurements. But if they're growing really slowly, that difference might be really small. And we're using pretty crude instruments to measure growth. So if we go out and measure the diameter of a tree at age 55, and it's 20.1 centimeters, and then we go out the next year and measure the diameter of that tree, and it says it's 20.3 centimeters, we probably don't have a lot of confidence in that number, because the resolution of our instrument is probably within the range of what the actual diameter growth of that tree was. So when tree growth slows down, those repeated measures are harder to do well. So sometimes, one way to adapt to that is to just use inventories every 5 years to estimate tree growth in these older stands, because the growth's really slow, but we can more with better precision and accuracy, well, with better accuracy, measure 5 years worth of growth than we can measure 1 year's worth of growth, just because of the limitation of our diameter tapes, for instance. Okay? So I just wanted to make that point. We're about out of time. I wanted to talk a little bit about juvenile wood and mature wood and about reaction wood, but I think that's a little bit arcane and not super relevant to mensuration. So just to keep us on schedule, y'all can come by and visit sometime, and I'll talk to you about juvenile wood and reaction wood, or take tree biology. Does anyone have any questions about what I've talked about? Okay. If anything comes up, drop me an email. My email's on the first slide. Russ and David, I really appreciate you inviting me. It's another chance to make contact with students, and I get to talk about tree biology, so I love that.
I'm glad we could make it happen.
Yeah.
Thank you, Tim.
Y'all take care.
Thank you guys. Thanks, Dr. Martin, Dr. Fox, Russ.